Okay, good afternoon, everyone. Welcome back. Hope you're doing well. Before we get started with talking about the lactose operon, do you have any questions at this time as we head into the last weeks of the semester? Okay. All right. So just so that I know, please type yes into the chat window if you can hear me okay. 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 Thank you so much. All right. Okay, gang. So, we'll keep doing this. And I will keep reminding you, the final exam has been released at this point, and I indicated that in my last email to you. The final exam for your course is going to be on Tuesday, May 12th. Tuesday, May 12th. Okay. And it will be a scenario where you have access to the exam from 7 a.m. to midnight on that day. And you can take the final exam during that window of time on Tuesday, May 12th. Okay. All right, gang, let's get into it here. I'm going to start sharing my screen with you again, and we're going to talk about the lac operon. Last time we mentioned that the purpose of the lactose operon is to allow bacteria like E. coli to metabolize the sugar lactose and use it as an energy supply to manufacture ATP. But we need to explain to you how this process is regulated, how the gene expression is regulated. Okay, so I gave you a bunch of background information last time, and we're going to continue here. What I'm going to do is we need to draw a schematic of the lactose operon in your notes. Okay, so I hope you have some colored pencils and things nearby that might be helpful today. But I'm going to start by putting in a schematic of the lactose operon, and for this, refer to the PowerPoint drawing. Okay, that we're going to do now, and of course you draw your own at home. Okay. All right. So here we go. Okay, so there are a number of schematics for the lactose operon already drawn in your textbook, and your textbook draws it multiple times to illustrate different points at different points during the conversation in the textbook to help you understand things, right? So for example, you have a schematic of the lactose operon on figure 1 on page 338. You have more schematics on figure 5, page 340. More schematics on figure 6, page 341. A schematic on page 342, a schematic on page 343. Basically, folks, all of these schematics that you see are kind of the exact same thing, but at any given moment on each page, the author of the book is trying to emphasize a certain aspect of the conversation. So we're not going to draw that many schematics, but we are going to draw an overview schematic so we can talk about this. All right. Okay, so we have here a schematic a schematic of the lactose operon. Okay, so all right, here's what we're going to do. Okay, so far I've drawn P hyphen cap hyphen 2 horizontal vertical bars, another P hyphen ADCY, 2 more vertical bars, P hyphen I hyphen P hyphen O hyphen L hyphen Z hyphen Y hyphen A. Okay, so this is our schematic of the lactose operon, and actually it's more than just the lactose operon. So let's be clear here what we're looking at. When we draw these symbols out, we are drawing a blueprint, a schematic of the gene sequences that we find along the E. coli chromosome. Okay, so what you actually have here is all of this is a section, all of this is a section of the E. coli chromosome. All right. And what we're going to have to do here is define every part of our schematic. Okay. And so let's begin that process. Okay, anytime you see a P, okay, the P's, these are P's for promoters. Okay, so the P's are the promoters. All right, so you recall the promoter is the binding target for RNA polymerase. Okay. All right. So remember, well, see, I need to bring up the chat window here so I can talk with you folks. Okay, just one moment. Okay. Okay. So at this time, remember our definition of an operon from our last conversation. An operon is a group of genes that are all transcribed from the same promoter. Okay? An operon is a group of genes transcribed from the same promoter. So I have a question coming through the chat window. How many operons are drawn on the screen? Okay, John, you say 1. Thank you. Malachi, 1. And okay, is that the consensus among all of you that we have 1 operon? Okay. Anybody else want to make a counter argument here? Because 1 is not correct. Okay. Ian is asking 4 with a question mark. That is correct. There are 4 operons here. Okay. Here's what I have. All right. I've got 4 promoters. 1, 2, 3, 4. Okay. Any of the genes that are transcribed from that common promoter are part of that operon. So that means I have this operon, I have this operon, I have this operon, and I have this operon. Okay? please understand that an operon is a group of genes transcribed from a single promoter. Please understand that that group could be a group of one, right? I've got one gene here, CAP, made from this promoter. I've got one gene here, ADCY, made from this promoter. I've got one gene here, I, made from this promoter. I've got 3 genes here for reasons that I'll explain next, Z, Y, and A, made from this promoter. Okay, so we've got 4 promoters, so that means we have 4 operons here. Does that make sense? Okay, so while this is the schematic of the lactose operon, I suppose it would be important to, to be very pedantic here and say schematic of the lactose operon and neighboring operons. Schematic of the lactose operon and neighboring operons. Okay. So, all right, let's define everything here on the screen. Okay, let's see. Please feel free to ask questions along the way, interrupt me, that's quite all right. CAP, okay, CAP. Okay, this sequence here, CAP, C-A-P. Folks, CAP is the gene for a protein called the catabolite activating protein. Okay, C-A-P, catabolite activating protein. So what we're saying in our schematic here is that this CAP gene is the stretch of DNA that codes, it's the stretch of DNA that contains the information that allows RNA polymerase to make a piece of CAP messenger RNA, which then a bacterial ribosome can translate into the actual protein that we call CAP. Okay? All right. Next, we have ADCY up here. ADCY. This is the gene for an enzyme called adenylate cyclase. Okay, adenylate cyclase, which is also known as adenylyl cyclase. Okay, you will find it in the literature both ways. Okay. Okay, ADCY is a gene for adenylate cyclase. Okay. The I gene up here, this is the gene for the lactose repressor protein. Gene for the lactose repressor protein. Okay. All right. So at this point, I'm going to stop and point out an important feature or a couple important features or characteristics. Okay. First off, you notice in the schematic that, let's see here, I need a good color here. You notice that, all right. that I have these vertical lines in the schematic, right? I have vertical lines here, vertical lines here. This is my way of telling you that these operons that are separated by the vertical lines are in fact not physically together in the same stretch of DNA on the E. coli chromosome. They are distantly apart from each other on the E. coli chromosome. They are not sitting next to each other. Okay? So separated on the chromosome. Okay, remember the E. coli chromosome is about 4.6 million nucleotide pairs of DNA in this gigantic circle. Okay, and so what we're saying is that the cap operon here is at one location of the circular chromosome, and the ADCY operon is at a physically different location. But even though these are at different locations on the E. coli chromosome, they all play an important role in regulating the lactose operon. Okay? All right. Any questions so far? All right. It's important that you appreciate, and I will put it down here. It's important that you appreciate, let's see, that all three of these genes, CAP, ADCY, and I, they are all constitutive genes. Refresh my memory from your notes. Type into the chat window, what does constitutive mean? What does constitutive mean, gang? What's that mean? Anybody? Okay, well, let me move us forward here. But you should find in your notes from last time that constitutive means that the genes are always on. Thank you, Malachi. Exactly. The genes are always on. They are always expressed. They are like a leaky faucet that does not turn off. Okay? All right, so these genes that are made from three different promoters that are part of operons that are in three different segments of the E. coli chromosome are they're on all the time. The cell has no way to turn them off completely. Okay? Questions? All right. All right, so if
control the lactose operon, or they make up the lactose operon.
Okay, thank you for the question, Hayden, and that tells me I need to clarify some things. So everything we have here is not part of the lactose operon, but is in part of multiple operons that all influence the lactose operon. Okay, so let me do this. Let me get another color here. Okay, so from here to here, we have the cap operon. From here to here, we have the ADCY operon. From here to here, we have the lac repressor operon. And from here to here, we have the lactose operon. Okay, so we have 4 operons. The first one's the cap operon. The second one is the ADCY operon. The third one is the lac repressor operon. And the fourth one is the lactose operon. Now, even though we're talking about the lactose operon, you have to understand that the lactose operon is regulated by molecules that are in fact made by other operons, not just the lactose operon. Okay, is that making sense, Hayden?
Yes.
Okay. All right, so let's continue here. All right, so the cap operon, the ADCY operon, and the lac repressor operon all code for proteins that are constitutively made. They are always made. Those genes are never shut off 100%. Now we're going to talk specifically about the symbols in the lactose operon. Okay, so let's see here. Let me draw a little separating line here. All right, so obviously P in the lactose operon is in fact the promoter. Okay, so I won't write that down because P is always promoter. O is a special DNA word called the operator sequence. Okay, so O is a DNA word for operator. Okay. It is not a gene. It does not code for anything. It is a regulation information hotspot, but it does not code for anything. Okay, so O is not a gene. Okay. L is something called a leader. A leader is a stretch of RNA that you typically find more in prokaryotic systems than you do in eukaryotic systems. That's just a general statement. You can find exceptions to that. But a leader is a stretch of DNA that is transcribed into RNA but never translated. Okay, so the leader is a gene in the sense that it is transcribed, it's made into RNA, but that leader is not translated into protein. Okay. Is made into RNA. It's made into RNA, but not translated. Okay. Now I'm pointing this out because we're going to be talking about the leader in the next system, which is the tryptophan operon. Truth be told, gang, for the lactose operon, even though there is a small leader present, it has never been identified as having any functional role. So it's kind of like your appendix. It doesn't do much for you, okay? So at least in the context of the lactose operon, it's fairly insignificant. Okay. All right. Let's continue here. Z is the gene that codes for a special enzyme called beta-galactosidase. Okay, beta, which is often abbreviated with the Greek symbol beta. Galactosidase. We will come in and potentially put in some functions of these molecules in just a moment, or type them into the Word document so it's nice and neat for you. Let me come over here so I have more space. The Y gene is a gene for an enzyme called permease. And the A gene is a gene that codes for an enzyme that is still a little bit of a mystery what this enzyme does, although I will talk about that in just a moment. But from a biochemical perspective, if you were going to characterize this as being similar to other known enzymes, well, the best we can say at this point is that it's an enzyme that resembles other enzymes that chemists know as transacetylases. So this is a gene for a transacetyl acetylase. Okay. All right. Questions so far? Okay. Now, there's one other aspect to our schematic that I want to put in here so that you have it in your notes. And it's the observation on figure 10 of page 345 that operator sequence is actually made of 3 independent locations. Okay, there's actually 3 independent operator stretches of DNA in this region of the E. coli chromosome. Okay, the very first one, and let me see, let me change my color so it stands out. The very first one is between the operator and leader. So right here is O1 for operator 1. O1 for operator 1. O2 is literally within the nucleotide sequence that codes for the Z gene. So O2 is here within the Z gene. And the third operator binding target is defined in between the LAC-I gene and the next promoter. So O3 is located about right here. O3 is right here. Okay. All right. Gang, any questions about the schematic at this moment? All right. Okay, so hopefully you can, you can have that drawing in front of you when you're studying, or as we're talking here, it can make some sense to you, and you have your textbook available, so you have all these resources that you can be thinking about content. So let me go back to the Word document. Okay, and let's take some notes on the schematic. Notes about the schematic, okay? All right, some things that I want you to appreciate. All right, so the CAP gene, okay, as we said, this codes for the catabolite activating protein, okay? Do you remember, I believe at the end of last class period, we were talking about the reality that if an E. coli cell finds itself in the presence of glucose and lactose, the E. coli cell will choose not to use the lactose because the E. coli cell prefers to use glucose. And we said at the end of the class last time that phenomenon where a cell can choose to use one sugar over another sugar is actually, it actually has a name, it's called catabolite repression. So here you have a gene that codes for a protein that's influential in catabolite repression. Okay. Okay, let's see here. This protein, CAP, is involved in the catabolite repression phenomenon. Okay. If I'm going too fast, let me know. All right. Next, the ADCY gene. Okay. Codes for adenylate cyclase, which again is also known as adenylyl cyclase. Okay. This enzyme, this protein, this enzyme is actually manufactured in an inactive state. Okay, so the gene is on all the time. It's constitutive, and we're constantly cranking out adenylate cyclase enzyme, but the adenylate cyclase enzyme is manufactured in a turned off state. Do you know what an enzyme is called when it's manufactured in an off state and has to be activated? Do you know what that is called? Oh, okay, Hayden. Thank you for trying with that. No, okay. No to Hayden and Caitlin, but thank you for trying. Okay. When you use the word inducible here, we're talking about a gene that is off and then it is turned on. But in this case, this gene is already on all the time, and it makes something that's in an inactive state and must be activated by other molecules in the cell. Okay, folks, an inactive enzyme is called a zymogen. Okay, an inactive enzyme is called a zymogen. All right. What else you should know about this is that when ADC, when adenylate cyclase, when the protein is activated, it's responsible for making something that's also known as a secondary messenger in animal physiology. Do you know what that is? Adenylate cyclase, when active, is responsible for making, can you answer that? Okay, we'll move the conversation forward. Gang, it's responsible for making cyclic AMP. Cyclic AMP, if you've had any coursework in the past where you were talking about hormones, cyclic AMP is a very common secondary messenger in hormonal regulation. Okay, but in this case, we're not talking about hormones of the endocrine system. We're talking about a bacterial cell. So adenylate cyclase is made in an inactive state, and based on circumstances, which I'll describe in a moment, becomes active. When it becomes active, it manufactures cyclic AMP. Okay? All right. Next, the I gene. Okay, the I gene. Let me write gene up here for ADCY as well. Okay, the I gene is, this codes for the lactose repressor protein. And as we mentioned, it's always on. It's a constitutive gene. And what's important for you to appreciate is that the lactose repressor protein's binding target is the operator of the lactose operon. The lactose repressor protein binds the operator sequence of the lactose operon. Okay, so its binding target is the O sequence that we put in your schematic. It binds the operator. Okay. The Z gene, which codes for beta-galactosidase. Well, this enzyme is responsible for the breakdown of lactose. if I'm being extremely anal, it's actually responsible for breaking down an isomer of lactose called allolactose. But we won't go quite there right yet, okay? So the beta-galactosidase enzyme is responsible for breaking down the sugar lactose. The Y gene, as in why the hell do I have to know all of this? The Y gene codes for the permease enzyme. Okay, this enzyme is responsible for bringing the lactose sugar into the cytoplasm of the cell. The permease enzyme facilitates the transport of lactose in the environment outside the cell through the cell wall and plasma membrane of the bacterial cell to get it into the cytoplasm. This enzyme is responsible for bringing lactose into the cell, okay, into the cytoplasm. Right? If the lactose is outside the cell, that's all great, but if I can't get it in, then that's not going to be good. I need it to be inside the cell to metabolize it and use it as fuel to make ATP. And then we have the A gene, okay? The A gene codes for the transacetylase. Okay, and this enzyme's function is a little mysterious, okay, but there is some evidence to suggest that this enzyme plays a role in breaking down metabolic waste products that arise from lactose metabolism. So as lactose is broken down chemically, there are some chemical waste products that are thrown out into the cytoplasm. And the leading hypothesis at this moment is that the transacetylase, its role might be in fact to chemically modify those waste products so that those waste products are not toxic and dangerous to the bacterial cell. So there's some evidence to suggest that this enzyme helps detoxify metabolic waste products produced during the breakdown of lactose. Okay? Okay. All right, gang. So, any questions at this moment? Okay, so recall from our conversation last time. I know I asked these questions to you. Okay, recall that there are two circumstances that must occur for the lactose operon genes to be expressed. Okay, you got them last time. That is that glucose must be absent. Glucose must be absent. If glucose is not absent, then catabolite repression will occur and the genes of the lactose operon will stay turned off. So glucose must be absent, otherwise the lac operon genes stay turned off, and this, it's important you understand the logic of this. This is an and. These two things have to happen together. It's not an or. So glucose must be absent and lactose must be present. Okay. So to understand this, Okay. Let me go back to the PowerPoint. Okay. Let me go back to the PowerPoint and let's see. I want to try to copy everything and put it on a second slide. Now, come on. Okay. All right. So let's see here. Okay. Okay, so I'm on the first slide here, and obviously you don't want to erase anything in your own notes, but I am going to erase some things here because I need more space here. Okay. All right. So when glucose is present, in the cell, that's going to keep the genes off, right? When glucose is present, that's going to keep the genes off. And how that works is kind of interesting here. Okay, let me get this down. Okay. All right. So let's see. Let me get my thoughts together here. For catabolite repression to work, okay. So let me switch that up. When glucose is absent, okay? When glucose is absent, that's going to lead to an activation of the adenylate cyclase enzyme. And if adenylate cyclase is active, it's going to manufacture cyclic AMP. Okay, so this leads to a production of cyclic AMP. So in short here, okay, let me get rid of the rest of this junk here. Okay. Okay. The take-home message here is that when glucose is low, cyclic AMP will go up. All right. Now, one of the pieces of the puzzle that we haven't shared with you yet, but is critical, is to appreciate that at the lactose promoter, okay, there is a cap binding region, okay? So if we were to take the promoter here and magnify into it, okay, so this promoter right here, if we were to magnify into it, we would actually find a binding site for the cap protein and RNA polymerase. Okay. Now, the cap protein is only capable of binding to this cap binding site if the cap protein is itself attached with cyclic AMP. Okay? Cap protein must be attached by cyclic AMP to bind the cap sequence that is part of the lac operon promoter. Okay. The cap protein must be attached by cyclic AMP in order to bind to the cap sequence of the promoter. So if I have cap protein, and let's see, if I draw the cap protein like this, and I take a wedge out of it, okay, and say that wedge there is cyclic AMP, where cyclic AMP binds. So I have cap cyclic AMP. That combination of molecules is what normally binds, this normally binds to the cap sequence of the promoter, okay? If cyclic AMP is not available and all I have is a naked cap protein, then the naked cap protein cannot bind to the cap sequence of the promoter. Okay, so we need cap and cyclic AMP in order to bind to the cap promoter. But if we do have this circumstance being true, okay, there's a lot of variables to get this gene regulation just right. So I know this is pretty involved. Okay. If cap cyclic AMP binds to the cap region of the promoter, when the cap cyclic AMP binds to the DNA of the promoter, it physically bends the promoter in such a way that it allows RNA polymerase to attach more firmly to the promoter. In other words, it helps RNA polymerase get a lock on it. If cap cyclic AMP binds to the cap region of the promoter, then RNA polymerase can attach more successfully. Okay. All right. So before I continue with this, there's a lot of stuff going on right now. Okay. When glucose is absent, think about it. If glucose is absent, that opens the possibility that we might want to use an alternative sugar, right? Because if glucose is absent, it's not available to be metabolized. So I might want to use an alternative sugar. So if glucose is absent, the cell turns on adenylate cyclase to manufacture cyclic AMP. The manufacturing of cyclic AMP is necessary to have a molecule that then can complex with the cap protein. So if I have cyclic AMP in the cell, that tells me that there's no glucose around, which means I might want to express the genes to metabolize an alternative sugar, like lactose. So when cyclic AMP is available, it binds to the cap protein, and then this complex of cap and cyclic AMP binds to the promoter region of the lactose operon, and it bends the DNA so that RNA polymerase can attach to that promoter. And if RNA polymerase can attach to that promoter, there's a good chance it can transcribe these genes, Z, Y, and A. Okay, questions right now before I go any further? Would it help to put this again down in the Word document? Is that a yes, John? Okay, all right, fair enough. All right, so let's go back to the Word document, and you have your drawing and your schematics in the textbook in front of you, okay? All right. Controlling the expression of the lac operon genes, okay? Okay, all right. We'll work at a couple different angles. We'll work at a couple different ways. If glucose is present, then adenylate cyclase is not activated, so no cyclic AMP is made. If no cyclic AMP is made, then there is no cyclic AMP to attach to cap, to the cap protein, okay? The cap protein cannot attach to the cap region of the lac operon promoter if it is not bound with cyclic AMP. Okay. So gang, let me ask you this question at this time. What molecule have we been talking about, coming through the chat window, what molecule have we been talking about that is a sensor for the presence of glucose in the cell? What molecule have we been talking about that is a sensor for the presence or absence of glucose in the cell? Okay, John, very good. Cyclic AMP, that's what I was hoping you would see, right? The cap protein is not the sensor because the cap protein is always made because the cap gene is a constitutive gene. But the substance that gets made is cyclic AMP. The only time cyclic AMP gets made is when adenylate cyclase is active. And the only time adenylate cyclase is active is if glucose is absent. Okay? Because if glucose is present, adenylate cyclase is not active, so no cyclic AMP is made. All right? So it is the presence or absence of cyclic AMP that tells the cell what it should be doing in terms of catabolite repression. Okay? It is the absence or presence of cyclic AMP that acts as a glucose sensor to inform the cell whether catabolite repression should take place or not. Okay. Let me say the same logic from the other perspective, okay? if you understand 3A, if glucose is present, then adenylate cyclase is not active, so no cyclic AMP is made. Okay, that is equivalent to saying if glucose is up, cyclic AMP will be down. Okay. Now I'm going to do the same logic from a different perspective. Same logic now from a different perspective. If glucose is absent, adenylate cyclase is activated, so cyclic AMP is made. If that's true, okay, if that's true, then CAP will attach to cyclic AMP, and the CAP cyclic AMP complex will attach to the CAP region of the lactose promoter, bending the DNA, allowing RNA polymerase to successfully attach to the promoter. Okay, so this is how the catabolite repression mechanism works. If glucose is present, adenylate cyclase is not activated so that there's no cyclic AMP. If there's no cyclic AMP, you can't bind the CAP protein. If the CAP cyclic AMP complex is not available, you can't bind to the CAP region of the LAC promoter, so the DNA is not bent and RNA polymerase cannot attach, so therefore the genes of the lactose operon cannot be expressed. I do have a video to show you all this, gang, but we have to walk through the logic first. So any questions right now? Okay. All right. This is one of those things, please understand, when we go through this, you are seriously looking at about 15 years of dedicated research that it took scientists to figure this thing out, right? This was not something they figured out in a weekend, okay? So this is many, many years of research that we're covering right now. Okay, so, right, the topic here is controlling the expression of the lactose operon gene. So statements A and B, okay, so let me complete statement B here, right? We said that if glucose is down, then cyclic AMP is going to be up, right? That's exactly the opposite here, right? I'm saying the same thing, two different ways from two different viewing perspectives. Okay. All right, so this explains how catabolite repression plays a role in keeping the genes of the lactose operon shut down or permitting them the possibility of being expressed. Okay. Now remember, if glucose is absent, that opens the possibility that the lac operon gene should be expressed, but it doesn't guarantee it, because the second thing that has to happen at the same time in the absence of glucose is that lactose must be present. Lactose must be present. Okay. So let me go back here to this horrible drawing. Okay. Okay. So let's say that glucose is low, cyclic AMP is up, which means cyclic AMP binds to the CAP protein. So CAP cyclic AMP binds to this promoter, which bends it so RNA polymerase can attach to it. Okay, so right now, I know the drawing is getting very, very ugly right now. Right now, in our scenario that we're talking about, we have RNA polymerase attached to this promoter because this cap cyclic AMP is also attached to this segment of DNA. Now, RNA polymerase, even if it gets attached to this promoter, it may not be able to physically access the genes Z, Y, and A. Well, why not? Can you answer that? Even when, coming through the chat window, even when RNA polymerase is attached to the LAC promoter, it may not be able to physically access the LAC genes. Why not? You have everything in your notes right now to answer that. Think about it, gang. Even if RNA polymerase is bound to this promoter, RNA polymerase may not be able to move through and transcribe genes Z, Y, or A. Why is that the case? Because they're separated on the chromosome. Well, thanks for trying, John, and I love the thinking, I really do. That doesn't happen to be the case right now. I mean, they are separated by a little bit of distance, but not a significant amount. That's not the issue here. What else, gang? Okay, Morgan, good job. The lactose repressor protein on the operator, right? Remember, this word, this O operator sequence is the binding target for the lactose repressor protein. Okay, so let me draw that in here as well. I know it's getting insane how many different molecules we have floating around, but this is the reality of it. Right now, I have this R protein, this repressor protein that's attached to the operator. LAC repressor. And it's always attached to the operator because the gene for the LAC repressor is constitutive. It's always on, which means it is always attached to the operator. So what is the ingenious way that Mother Nature decided we could remove the LAC repressor protein from the operator? Do you know? How do we remove the LAC repressor protein from the operator? What's going to be the thing that makes that possible? High levels of cyclic AMP. That's an interesting idea, John, because obviously if there's cyclic AMP, we can bind to cap, which can then combine to the promoter, and maybe that twists the DNA enough to eject the repressor protein. That's a good thought. It really is. But that's not what Mother Nature came up with. What else? Okay, what is the second circumstance I need to be true in order to express the LAC operon genes? I need to have no glucose, and I need to have gang, I need to have lactose present. There you go, Ian. Good. Okay. So when lactose is present in the environment, lactose has the ability to physically bind to the lactose repressor. So if I have lactose sugar present, these two things will bind. And when lactose binds the LAC repressor protein, it changes the shape of the protein so the protein cannot keep its grip on the DNA. So then the protein literally falls off the operator sequence. And if that protein, the repressor protein, falls off the operator sequence, then RNA polymerase that's been allowed to attach to the promoter can physically walk through here and transcribe genes Z, Y, and A. Okay, let's go back to our notes here. This is pretty intense stuff here, okay? All right, so number 3, controlling the expression of the lac operon genes. Letters A and B are telling you the same thing from a different viewing perspective. Okay. Okay. When note B is true, we have the possibility of expressing the lac operon genes as long as lactose is present while glucose is absent. Okay. How this works is lactose sugar binds to the lactose repressor protein, changes its shape so the lac repressor protein detaches from the DNA. Do you see, gang, that while the lac repressor protein is physically attached to the operator sequence, it acts as an NFL linebacker to physically block any motion of RNA polymerase. If RNA polymerase tries to go and express gene Z, Y, and A, it will physically run into the repressor protein and be blocked if the repressor protein is present. So to prevent that circumstance from happening, lactose sugar binds to the repressor, changes the repressor protein shape so it falls off the DNA. Then RNA polymerase is not going to hit the NFL linebacker. It's not going to get blocked by the protein as it moves across that segment of DNA to begin transcribing gene Z, transcribing gene Y, transcribing gene A. Okay. With the lactose repressor protein removed, RNA polymerase can now access genes Z, Y, and A. Okay, questions right now? All right, I think we've got it, okay? It is a complicated scenario, but this is one of the best understood genetic regulatory circuits in all of science. We know this better than just about anything because it has been studied for decades. And I assure you that this knowledge is used every day in modern genetic engineering laboratories to do things like force bacteria to manufacture insulin and recombinant antibodies and all kinds of things we like to use in modern medicine. Okay, so let me pause and go to the video to try to consolidate all of our knowledge at this time, all the things we've been talking about. Let's see this video to make sense of everything happening right now. Here we go. Sorry, this isn't high definition version, but they don't have a high def version, but I think it'll still work for us. So here we go.
The tryptophan operon, trip operon.
Wrong video. Let's do this one. The lac operon. Here we go.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the LAC operon. The unit consisting of the LAC promoter, LAC operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the LAC operon. Located near, but not in tandem with, the LAC region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the LAC repressor. In the absence of lactose inside the cell, the LAC repressor protein is active. In its active state, the LAC repressor recognizes and binds to the LAC operator site. When the LAC repressor is bound to the LAC operator site, RNA polymerase is prohibited from recognizing the LAC promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed.
Gang, in your textbook, on page 345, figure 11, it shows you the crystal structure of the lactose repressor protein, and it shows you what it actually does to the lactose operon DNA when it binds. It actually causes a loop to form. That's shown to you on figure 11. So again, it reinforces the idea that proteins modify the structure and shape, the shape of the DNA, allowing it to turn on or turn off the expression of genes. Okay, I will continue.
LAC-Y and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the LAC repressor. This causes a conformational change in the LAC repressor protein that prevents it from binding to the LAC operator site. Inactive LAC repressor is unable to bind to the LAC operator site. Without an active LAC repressor bound to the LAC operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the LAC operon enables the bacterium to efficiently transport lactose into the cell and metabolize it. The expression.
Okay, gang, so this video shared with you half of the regulatory scheme for activating expression of the genes. I'm having a hard time finding a video that covers both aspects of regulation. There used to be an old video online that seems to have disappeared from YouTube. So if I find it, I'll share it with you. But obviously this video showed you how the lactose repressor is used to regulate the expression of the LAC operon genes, but it did not cover with you the catabolite repression mechanism, which again requires cyclic AMP and CAP to be bound to the LAC promoter. That will only happen if cyclic AMP is available. That will only happen if adenylate cyclase is active, and that will only happen if glucose levels are low inside the cell. Okay, what questions do you have at this time? All right, give me a minute or two. Let's see if we can poke some holes in your understanding, because I really need you to understand this, which is a strong indication that this is going to be a major deal on your upcoming final exam. Okay, so here's a question for you. If the genes of the LAC operon are being expressed, how do we turn them back off? What do you think, gang? Okay, how do you think we can turn the genes for the LAC operon off if they're actually being expressed? What could we do to turn them off? Add glucose. Okay, I like that. Very good. Okay, so we could add glucose. Okay, help me complete my statement here, Hayden, or any of you. If I add glucose, what's that going to do to adenylate cyclase? If I add glucose, what's that going to do to adenylate cyclase? It's not going to stop production, it's going to stop activation. There's a difference, right? You can have a molecule be present but not be on. So you have the right idea, but it's not a production thing, it's a deactivation thing. If we add glucose, we deactivate adenylate cyclase, which means what happens to our cyclic AMP level? Does it go up, down, or stay the same? Will we, okay, very good, John. Okay, we're not going to make any additional cyclic AMP. That's what I'm trying to get out of you, okay? So cyclic AMP, no new cyclic AMP will get made, okay? So, you know, there might be some remnant cyclic AMP that hangs around for a while before it gets degraded by the normal chemistry of the cell. But once that's degraded, no additional cyclic AMP is going to be made in the presence of glucose. And if that's true, will RNA polymerase be able to bind to the promoter? Will RNA polymerase be able to bind to the promoter if all of a sudden there's no cyclic AMP around? Correct, Ian. Why not? Why will RNA polymerase not be able to bind to the promoter if there's no cyclic AMP around? Anybody? If there's no cyclic AMP, why can RNA polymerase not attach to the promoter? Correct, Hayden, because RNA polymerase will not be capable of binding to the promoter because there will be no cap cyclic AMP bound to the promoter, twisting it so that RNA polymerase can attach. That is correct. So let's finish the logic here. No new cyclic AMP will get made, so RNA polymerase cannot attach to the promoter because the promoter DNA is not bent by the presence of a cap cyclic AMP complex. Gang, is this making sense? Okay, all right. So we could in fact add glucose to turn the lac operon off when it was on. What else could we do? I mean, realistically speaking, if the lac operon genes are on, doesn't that suggest to you that that bacterial cell is making the enzymes from the lac operon because it wants to eat the lactose in the environment? So if the bacterium is actively eating the lactose, can you imagine a scenario where the bacterium eats all of the lactose? It's kind of like, yes, you have a bunch of food in your refrigerator, but eventually by the end of the week, you're going to eat most of that food. And unless you go back to the supermarket, you're not going to have any more food. So the point of it is, if the lac operon genes are being expressed, that means the bacterium is actively eating the lactose sugar. So lactose levels eventually drop to 0 because the enzymes of the lac operon genes are metabolizing the sugar. If the lactose levels drop to 0, what's going to happen with the lac repressor? If the lactose levels drop to 0, what's going to happen to the lactose repressor? Anybody? Hayden, you say it falls off. What do you mean? You mean the repressor falls off the DNA? Is that what you mean? If the lactose genes are actively being expressed, the lactose repressor is not attached to the operator. But if lactose levels drop off, if the lactose levels go to 0, what's the repressor protein going to do? Gang, do you see that if the bacterium eats all the lactose, then all of a sudden there's no lactose to attach to the repressor to keep the repressor off the DNA? So what's the repressor going to do? Okay. This is tough stuff, and it's going to take a while for you to think through this on your study time, but I'll give you the answer right now. If the lactose levels drop to 0, the repressor protein is going to revert back to its original shape that allows it to reattach to the operator. And if it reattaches to the operator, then the repressor protein is once again going to be an NFL linebacker physically blocking RNA polymerase. So we will turn off the expression of the genes of the lactose operon. Okay, in your notes, if the lactose levels drop to 0, lac repressor protein reverts to its original shape and reattaches to the operator sequence on the DNA. This blocks RNA polymerase from any further gene expression. Okay. All right, gang. I know we're over time, but what questions do you have at this moment? Okay. Spend some real quality time making sure you deeply understand the lactose operon. It's important, not just to be a biologist, but to do well on your final exam. It's important. And next class, when we start up, I'm gonna start talking about the tryptophan operon because it is a point of contrast to the lactose operon. So I'm hoping that next class we can cover the tryptophan operon in maybe 20 to 30 minutes. I'll field any questions you might have, and then we'll be on to chapter 13, believe it or not. Okay, because we've got to get through the content so that you have what you need to study. Okay, do you have any questions? All right. Well, gang, I'm going to take a mini break. I'll be back in about 10 minutes if any of you have questions or want to talk about anything. Otherwise, keep working hard. Try to stay focused. I know it's challenging. Everybody's got their own personal concerns right now, but try to focus so that you can finish the semester strong. So I'll see you in 10 minutes, or if not, have a nice weekend, and I'll see you next week, okay? Take care.